Ah, okay, great. Thank you for that introduction. Yeah, so I am happy to answer questions. But one of the things that you're going to find with my dissertation work is that these are sort of disparate scientific stories or systems, all sort of guided by a overarching set of questions. And so we'll sort of switch gears midway to a completely different story. So we're going to have sort of to do some shifting of focus, but I think it will work. Let's see here. There we go. Okay, there, great. So before I get going, before I get going, I want to thank the people that made this work possible. I have a bad habit of like leaving this to the end, and then I have like 10 seconds to say thank you. But so my wife and my parents were instrumental in sort of pushing me and emotionally supporting me in this really chaotic journey. My advisor, Laura, my lab mates, my many collaborators across the globe and the United States, the Organization for Tropical Studies, they run the Las Cruces Field Station where most of the field work took place. It's a really great place to do field work. Super posh. I highly recommend going there if you get the chance. North Dakota State, of course, for taking me on as a grad student, and the NSF funding that made this work possible. Okay, so I'll just use this. So the broad questions that sort of underlie this work are there predictable ways that fungi move through what I call mosaic landscapes? And are there management choices that can retain or restore fungal biodiversity in areas where agriculture and forest meet? Both within the context of present agricultural land and restoring forest on what was once agricultural land. And so let's begin by thinking about the story of tropical forests. This preemptively. So talking about tropical forests, the story of tropical forests is frequently told within the context of biodiversity, complexity, and immensity, the language of tropical forests housing a majority of the terrestrial biodiversity on Earth. But it's frequently told in the language of being under threat through many different interrelated anthropogenic pressures, urbanization, timber and mining, and most acutely agriculture. So agriculture is responsible for a lot of land use shifts in the tropics, and there are good reasons for that. I think it's important to think about those within the context of these questions. So in the tropics, where a lot of these forests exist, there's a strong tension between the ability and desire to conserve your ecological heritage and the ability to develop your country and your economy. And in many places where these extremely biodiverse regions are, many people still live in poverty. And a means of development is the incentivization of agriculture and global trade of those agricultural products. And so in places like Costa Rica, what has resulted is a sort of incentive structure that both makes agriculture easy to enter into, but also incentivizes things like harboring biodiversity on your farm. You get a credit for having a forest fragment, for example, or you can actually get paid for harboring biodiversity. So there's these tensions in the legislature itself that have produced essentially an enormous amount of fragmentation in the country. So what you'll notice here is that it's not just sort of clear cut. There's a huge number of sort of tiny fragments located within a mosaic of agricultural and forested land. And so that's essentially the system that we're working in. This is in southern Costa Rica, and that sort of little highlighted area is the biological station. Most of our sites were in this region. I think all of them actually were within this sort of region. So what this creates are managed natural interfaces, for lack of a better term here, where organisms like fungi, spores, or birds can move across ecosystem boundaries. Say you're sporulating in the coffee, that can blow into the forest and vice versa. And so ultimately, this is what I was concerned with in my PhD, this idea of ecological spillover, the movement of organisms and their associated functions across landscape or ecosystem boundaries. Now, ecological spillover is not new. This is has a rich history, but the most of the work on this in the past has been sort of agronomically framed, at least in the context of agriculture and natural interfaces, where your crop system, where the natural system adjacent to your crop system can be thought of as a reservoir for things like herbivores, pathogens, pollinators, and predators that can move into your coffee and influence your crop system. And very little work has been done relative to that in order to understand the capacity of the opposite, how a managed system can influence an adjacent, quote unquote, natural or less anthropogenic ecosystem. And so that's really the inversion that we wanted to make for my project with foliar fungi. And so when thinking about foliar fungi, we're talking oftentimes about an organism with a host. So these are things that are living on and inside of leaves. And so we have to combine our thinking of both ecological and zoonotic spillover to think about this concept. So zoonotic spillover is when we think about an organism jumping from one host species to another. And this increases in likelihood, generally speaking, the more dense or abundant that host is in a given area and whether or not there are close relatives nearby. So in the case of COVID, for example, you had a lot of sort of mammals in these markets and in close proximity to us. So essentially, that increases the likelihood of zoonotic spillover, whereas ecological spillover is oftentimes mediated by proximity. So if that boundary between your ecosystems or your two different biotic landscapes is small, then the likelihood that organisms can move across them goes up. And so we have to fuse these two ideas when thinking about fungi. So let's say you have something in the forest and it's sporulating and it blows into your coffee. It has to move in both physical and phylogenetic space. It has to move from forest or field to forest, and it has to move from coffee to some other compatible host. And so ultimately what we were looking for was this pattern of spillover that would be indicative of something moving from coffee to forest in the way we might expect for a foliar inhabiting fungus. And so what we're looking for is pressure, propagule pressure from coffee to forest, sort of greater abundances of these things sporulating in the air into the forest, and that sort of reflected in the phyllosphere of these leaves. And if we think about the relatedness component, we would expect this trend to be most sort of dramatic on close relatives of coffee, like this Psychotria popigiana, which is in the Rubiaceae family, which is the family coffee is in, compared to this Miconia, which is outside the family Rubiaceae. It's much more distantly related. And so we combine all these ideas. This is essentially what we're looking for. And coffee is a great model system to think about this because you get the proximity component. In Costa Rica, a huge amount of coffee is owned by smallholder farmers who have small fragments of forest on their land. And so you have the density component. Coffee is a monoculture. It's really densely packed. And you have the relatedness component. So while coffee is not native to Costa Rica, many native Rubiaceae exist in the forest understory. And so we can sort of ask these kinds of questions. Okay. So what kinds of questions are we asking? I kind of gave it away, but we're essentially asking, do coffee plantations act as reservoirs for coffee-associated foliar fungi that can affect adjacent forest plants? Ultimately, I suspect that they can, and this will be evidenced by these higher abundances in the air and on leaves near to the coffee forest border. And we can kind of fuse this idea with the second question of whether or not close relatives of coffee are more likely to have a more sort of steep spatial trend here. Okay. So in order to test this, the way we set it up was we partnered with coffee farmers in the Cotobruz region of Costa Rica, and we set up these transects at 8 sites. So each farm had a forest fragment directly adjacent to a coffee field. And the field had to be big enough to be able to run this transect 100 meters into the field without being 100 meters from an edge. And similarly, the forest fragment had to be that large as well. So you can get like, it's not as though you're going towards some opposite edge. If that makes sense. And what we did was we sampled leaves at 150, 25, and 10 meters in both directions from the border, in the coffee field and in the forest. And then we set up these passive airborne spore samplers that we built to characterize the airborne propagule community. The way that they work, they're modified Wilson and Cook samplers where we essentially created this sail that orients it based on wind so that the intake tube is always facing the wind. And so we set these up at 50 meters in the coffee at each site to get a sense of what's in the air there. And then we set up 2 at the border, one facing the coffee and one facing the forest. So these ones can't rotate. And this is characterizing what's blowing directly from coffee into the forest edge and directly from the forest into the coffee. And then at our different leaf sampling locations, 10, 25, 50, and 100 meters into the forest. And for our leaves, we had sort of a dual data pipeline. We were interested in disease, and disease is something that you often can see. And so we wanted to do some classic sort of plant pathology image data damage surveys. And so we photographed all of these leaves, and I calculated the proportion of leaf that was damaged, and then I morphotyped different damage types on the leaf. I did a lot more work with that data set than I'm going to talk about here. I'm happy to answer questions about that approach. But essentially, you know, I morphotyped different kinds of herbivory, different kinds of fungal disease, bacterial diseases, and basically created a presence or absence matrix of these different disease types. And then, of course, we characterized the fungal communities of these leaves using metabarcoding of the ITS2 region. And so what leaves are we actually sampling? Well, ultimately, in the coffee, we're sampling coffee. So coffee is the only thing that's really there. There are some shade trees, but it's a coffee field. So we're sampling coffee. Hope I've made myself clear there. In the forest, on the other hand, we wanted to both capture a number of different species and a number of different species with relation to coffee. So at each distance, this is going to get a little bit difficult to describe, but at each distance, we were sampling individuals from 3 Rubiaceae species and 2 non-Rubiaceae species, so at 10, 25, 50, and 100 meters. And we attempted to keep these species consistent at each of these distances, at least at the site. This is extremely difficult to do in the tropics because it's super biodiverse, and you're not going to find 3, 5 individuals of each species at each distance, but we did the best that we could. And for the most part, I think we actually did a pretty good job. There are like 3 of these species that are super common, so it's relatively easy to do this. But anyway, to summarize, we had 1 leaf per 4 individual coffee plants at our 4 distances, and then in the forest, we had 1 leaf by 3 to 5 individuals, depending on what we could find, of these 5 different species at our 4 distances, totaling essentially 938 leaves that I was taking pictures of and working with. Okay. So with our image data set, essentially what we zeroed in on was leaf spot. So this is a very common disease symptom. It's caused by a number of different kinds of fungi, but we were noticing a ton of it in the coffee. And so hoja de gallo is a coffee pathogen, a disease caused by Mycena citricolor, which is a Basidiomycete down there, and it's a devastating disease for coffee growers. And so we were particularly interested in that disease, and we had thought that maybe we were capturing a Mycena outbreak here. And so we focused in on that in terms of the visual data. And then for our molecular data set, we did not have infinite money, and we were sampling a lot of other stuff for this project. And so we pooled our individuals per species, per distance per site. So we had like a composite molecular sample of 3 to 5 individuals of a Psychotria propagiana at 10 meters at a given site. Hope that is clear. And so for airborne fungi, on the other hand, essentially we left these out just to passively collect fungi in the air for 4 months. We'd go out to each site once a month and replace the tube, or the sort of bottle, I should say, and we bring that back. We rinse it out with sterile water. We'd run that through a membrane on a vacuum pump, and then we'd have these membranes that we could use to characterize that had all the stuff that was in the tube or stuck to it. And so we had these composite samples of the sort of aggregate propagule pool in the air at these different distances. And just to clarify, in our modeling approach, we wanted to avoid pseudo replication, given that we're like sampling the same exact location. And so for the sake of the models, we aggregated all these months together to think of it as just like the total propagule aggregate at that distance at that site for 4 months. Some other wings of the project that I'm not going to go into here, but we also wanted to characterize biotic spillover. So another grad student and I, we misnetted birds in the coffee at the coffee forest border, and in the forest we swabbed the birds, and then we characterized what fungi were on the birds, what they were moving around. And then Diana is a current graduate student at NDSU, Diana Vargas Hernandez, and she is working with some of the endophyte isolates that I got from these rubiaceae leaves and is testing them for antagonism against Mycena citricolor. She's doing these different antagonism assays. Okay, so in a nutshell, giving you a lot of sort of setup for some of the results here. We're looking for a decline of coffee-associated fungi in the forest, both in the air and on leaves, and any kind of hallmark signal of relatedness that we might expect, given that these fungi need to find compatible hosts. And so let's begin with the visual data. It's sort of the most intuitive. This is the proportion of leaves with leaf spot. And the first thing you'll notice is that there's a lot of leaf spot in the coffee. It's like over half the leaves we're sampling have this disease on them, or this lesion type on them. There's much less leaf spot in the forest relative to that, but we do see this sort of decline among forest rubiaceae plants in the sort of odds that you're going to find leaf spot on a rubiaceae leaf. It declines with distance into the forest, or distance from the coffee forest border. And we don't see that in forest non-rubiaceae leaves. So what we're seeing here is exactly the kind of signal we were potentially expecting for a sort of coffee-mediated spillover of an organism that is in targeting sort of rubiaceae or is compatible with rubiaceae. And some collaborators also at 4 of our sites set up these 1 by 1 meter seedling plots, where they did this at 5 meters and then 10 meters thereafter to 105 meters in the forest. And they characterized every plant seedling that was in that 1 by 1 meter plot. And then they calculated the proportion of that plant seedling that was damaged by fungi. And then they built this model that was able to leverage phylogenetic distance from coffee and physical distance along the coffee forest border. And what they found was this interaction, essentially the same interaction that we're seeing with the leaf spot, whereas you see this steep decline of fungal damage from the coffee forest border into the forest among close relatives of coffee, but not in more distant relatives. Okay, so when we think about our molecular data set, so pairing that with this sort of visual data set, I think one of the very first approaches that I took was, okay, are there any just like groups that are super abundant in the coffee that we might be able to just see a signal in this entire taxonomic group? And the strongest candidate for this was Pleosporales, the order Pleosporales. It was accounted for about 40% of our reads in the coffee, and it was like triple the richness of any other order in the coffee. So we thought, okay, maybe this is like a good order. We might see a signal housed within this order itself. And so when we modeled Pleosporales relative abundance by distance along the transect with a beta binomial mixed model with low jet length, what we found was this striking decline in Pleosporales relative abundance on these leaves with distance along the transect. And this was robust to sort of different subsets of distances. It wasn't just an effect of sort of the coffee having really high abundances and the forest was low across the board. It wasn't leveraged out by that. And what we found in the forest was that this is largely driven by this decline in Pleosporales relative abundance in close coffee relatives in the rubiaceae. And it wasn't present. That sort of spatial signal was absent from the more distant relatives. And then when we looked at the airborne samples, we're comparing, we see the same sort of spatial trend. We're seeing this decline in relative abundance of Pleosporales in our airborne propagule total a long distance from the coffee forest border. Finally, we wanted to actually ask about particular taxa, not just a broad taxonomic group. So I built a generalized joint attribute model, which is a modeling approach where you can model a bunch of different response variables jointly. In our case, we used OTUs with different predictors. And so in this case, we had phylogenetic distance from coffee on the host plant. We had physical distance, we had site. And essentially what we found was that okay, let me back up. So this modeling approach has some limitations in terms of computational requirements. And so I couldn't just run the whole matrix on this model. So what we did was we took the approach of let's look at the 1,000 most abundant OTUs in coffee and see what they're doing in the forest. And first we filtered out anything that didn't appear on at least 10 forest samples. So when we did that, we were left with 836 OTUs. So what that shows right away is there's a broad sharing of these taxa across the boundary. But among these 836, what you're seeing is about 10% of them show this decline as you become more distantly related from coffee. So there's a broad suite of taxa that appear to preferentially associate with close relatives of coffee. And among this, we found 10 taxa or 10 OTUs that had that same interactive effect that we saw in the visual data set, where that effect of distance goes away when the host plant is more distantly related to coffee. Several Diademalaceae, and this is interesting, so that's a really diverse family, but there's many leaf spot pathogens of coffee within that family, including Phoma, and so these are some of the candidates that we found. And so ultimately, some takeaways from this work is that field to forest ecological spillover can operate, it appears, as a sort of cryptic edge effect of fragmentation. So this could potentially disfavor close relatives of the crop plant over time, if there's an effect on fitness, for example. And I think we have some evidence from the disease symptoms that might actually be the case. And there's some follow-up work being done here on plant community structure to see if that's actually borne out yet in these plant communities in the forest. Okay, so we're going to switch gears now from thinking about coffee in relation to adjacent forest to think about how do we restore a forest on what was agricultural land. And this was a close collaboration with Estefania Fernandez at the University of Missouri, St. Louis. So as we learned last week, restorations are attempts at particular goals. They're goal-oriented exercises. And so in order to do that, you make an attempt at a goal. You have metrics to determine if you were successful. And you determine if you were successful or not based on those metrics, and that can inform future practices, classic restoration, conservation ecology work. And traditionally, the way that this has been done for forest restorations, a common approach is to take a given restoration and look at the bird, mammal, plant communities, insect communities, and compare them to a nearby reference forest that hasn't been cleared and see if they're similar. And if they are similar, you might be able to determine that your restorations was more successful than, say, other approaches that don't attain that level of similarity. But conspicuously missing from this are fungi that are capable of degrading wood. So oftentimes when these kinds of restoration ecology projects are undertaken, they're looking at invertebrates, they're looking at frogs, they're looking at birds and plants, but they're not looking at any sort of saprotrophs. And I think that that's a pretty steep omission when thinking about a forest, which is in large part made up of these huge recalcitrant carbon pools. And so what I was just interested in is, does the restoration approach you take matter in recruiting decomposer fungi and decaying wood? And so there are different kinds of ways to go about restoring a forest. You can let it naturally regenerate on its own. You can fence off the pasture land and just let things recruit on their own. You can actually go in and plant seedlings to accelerate canopy closure and sort of supercharge the restoration process. And if you were comparing these two approaches, you'd have a nearby reference forest, and you could compare things like plant community, diversity, leaf area index, a bunch of different metrics you could potentially use. And this is exactly the system that we had. So Zach Zahawi and Karen Hull have these really wonderful long-term experiments in Costa Rica where they've done a natural regeneration and a plantation and have these reference forest fragments at 5 different sites. And they've been monitoring them for close to 20 years to assess which approach is better. But I'm also interested in how fungi disperse into these logs. When I entered graduate school, I was really keen on thinking about arthropods. And so one of the things that you'll notice in these logs, if you ever go and sort of play with decaying wood, is that there's all kinds of stuff sort of living on and in them, all kinds of invertebrates. And so spores can arrive from the air, but they also might be brought in on insects or in the mycangia, for example, or in the gut of these wood-boring arthropods. And so what I really wanted to know was, to what extent is this kind of dispersal relevant to these logs in these forest restorations? And so in order to assess this, we had these decaying logs that we deployed into the field. One was open to the environment, and 2 had this caging treatment where we had this aluminum mesh that made them less accessible to large arthropods. And we did have a third treatment in here. That's why there's 2 caging treatments where we drilled holes into the logs to imitate sort of the boring of wood feeding insects. But I'm not going to really go into that, as there's a lot of we don't really know if that approach did what we wanted it to do. We had some conflicting data with our arthropod surveys, and so we're just going to think about this as sort of a caging treatment. And so I just want to know which approach is, if any, better. And I suspected that plantations would operate better here because they're accelerating canopy closure. We have a lot of data on these sites already, and things like canopy cover in the plantations is more like a reference forest. Litter inputs are the same story. And then to what extent do arthropods bring fungi to these logs that otherwise wouldn't arrive? And I suspect that they do bring something, so that if we restrict their access, those things are not going to get there, and we'll see these reductions in alpha diversity. So we had these 3 log types and our 3 plot types at 5 sites, and we left them in the field for about a year. And what we found was essentially what I had expected. Plantations and reference forests were indistinguishable from each other in terms of beta diversity, but the natural regenerations were distinct from both of these kinds of plots. And our control logs were distinct from the 2 caging treatments, but the caging treatments themselves didn't differ from each other. And these 2 treatments didn't interact with each other. So this appears to be sort of these effects seem to be operating separately. In terms of fungal richness, what we found was that effectively the restoration plantation in the control logs, which are those blue lines there, they achieve sort of levels of reference forest richness. And when we caged them, we saw reductions in richness. But when we looked at the natural regeneration, we saw both that it didn't achieve sort of reference-like levels of richness in the control logs, and that this caging didn't seem to have any effect. And so ultimately, I think we have evidence here, just foundational evidence to support the idea that in this system, plantations are better at recruiting reference forest-like communities. And that arthropods do seem to bring fungi that otherwise don't arrive, but that this is contextually dependent. And I have some ideas as to why that might be in this given system. I can talk about those later, but essentially the natural regeneration is a much more open system. There's a lot less canopy cover, there's a lot more wind, and so these sort of windborne dispersal might actually just flood out any sort of influence of arthropods in that restoration. Why care? So one interesting finding that we found is that in our reference forests and our plantations, we were getting good matches for this particular Fungus G-Sternea davidii, which has been proposed as a species of concern to the IUCN because it's associated with declining tropical forests. We didn't detect it in the natural regenerations. And so ultimately, if restoration choices impact our ability to recruit declining taxa, then if we don't sort of establish this foundational work to understand which approaches function better, we're doing restoration in the dark, and we have very limited ability to sort of optimize our approach for given goals. And so it makes this work sort of worthwhile, in my view. All right. And finally, we're going to take a huge left turn, admittedly, and we're going to talk about buffalo fish. This is not related to my PhD work at all, but everybody loves a side project. And it is, I think, a really nice, interesting scientific story that I was lucky enough to be a part of. And I think it would be a nice way to close this talk. So this is me, I think, in 2010 or 2011, with a big mouth buffalo fish that I caught in a canoe with my dog. And that's Birdie. Birdie was a very close friend of mine. And okay, so these are remarkable fish for a number of reasons, but one of the things that sort of helps contextualize this work is that my brother and I would fish all the time when we were growing up. And we were really interested in this particular fish because every once in a while you would see them spawning in the rivers or see one swimming in the lake, or you might catch one by accident on rod and reel, but there was very little information available about them, and they were pretty mysterious. And so my brother, fast forward a few years, my brother Alec was in graduate school at North Dakota State, and he decided that he wanted to try to age these fish using otolith thin sectioning. So those of you that don't know, this is an approach where you take the calcium deposits or the ear stones of fish, you thin section them, and this accretes over the course of a lifetime. And so you can count the annual accretions and get a sense of an accurate estimate of the fish's age, similar to sort of dendrochronology work in trees.
Can you eat them?
Can you eat them? Yes. So commercial fisheries for these fish exist, and we did eat many of these fish and canned them, but it's not a widely targeted fish for food, and there's a lot of that's a whole other story that we can talk about if we have time. But essentially, what my brother discovered was this remarkable longevity to these fish. So it was thought that these fish maybe lived 20 to 30 years, but what he found is that these fish were regularly living over 100 years. So the oldest one from this study that he first published in 2019 was 112. So that was the estimate for how old this fish was. And so this totally was a paradigm shift in the life history of our understanding of the life history of this fish. And it set off my brother Alec's career, and he's been doing this kind of work with understudied fish in the Midwest at the University of Minnesota Duluth ever since. And he's, I think, on the Zoom today. So if you have questions, I think he might be able to chime in. And so anyway, fast forward, and now I'm in graduate school, and I get a call from my brother, and he says that there's these anglers, there's these fishermen in Arizona near Phoenix who are catching buffalo fish. They read my paper and they want to know if this fish is as old as the fish they're catching are as old as the ones that we had aged in Minnesota. And that was surprising for a number of reasons. The first is that buffalo fish are not supposed to be there, so that's well outside their native range. But what we found pretty soon thereafter, what we discovered was that there was, in 1912, Roosevelt Dam was constructed, and it created Apache Lake near Arizona, and a chain of lakes, essentially. And in 1918, we have records of 420 buffalo fish being transported from Fairport Biological Station in Iowa to Roosevelt Lake in Arizona in an attempt to establish a commercial fishery there. We have no idea if that was the only transport. There may have been other sort of undocumented transports, but essentially we have this small set of fish that were stocked artificially in this reservoir. And now in today, people are still catching them in Apache Lake. And so these group of anglers, a few of them are pictured here. That's Stuart Black in the middle. He was the sort of lead sort of organizer of the fishing group. They wanted us to come down and help age these fish to see how old they were. And so he established a fishing tournament. He got all these different people who are fascinated by these fish to come and fish exclusively for these fish over the course of a weekend. And we would age, or they would donate any specimen they caught to our cause. And we could age them and get a sense of how old these fish were. And these are a few of the fish, or this is actually one fish that's been caught several times by these anglers. So there's a number of recaptures that indicate that potentially this is a relatively small population. And so ultimately, what we found, okay, let me just, I think I have time, so I'm just gonna, the overall like pie in the sky dream of these anglers was to see if they could catch, somebody could catch one of these originally stocked fish. So that was like, that was the hunt for them. It was like everybody's goal was to catch that fish. And given how old these fish can live, it's not impossible that can actually happen. So this is not exactly a natural system, but it's what we were working with. So we went down there. Apache Lake is a really beautiful place. So if you ever get the chance to go, I highly recommend going there. It's like a really beautiful lake that is fun to be at, just in general. So what we found was that these fish are extremely old. So the vast majority of them are old. There's actually representatives from all three species of this fish that are found in the United States. Smallmouth, bigmouth, and black buffalo. And we have individuals from all three. And so what this first established was that all three of these species can live to be around potentially 100 years old. And most of them are between 80 and 108 years old based on our estimates, right? And so what this illustrates is something pretty remarkable, that potentially these fish are extremely old in this very, very different system than they're evolved to live in. And B, there does, unless we're missing some part of sort of the behavioral story, there appears to be broad long-term recruitment failure in this fish population. And that is also seemingly the case in parts of Minnesota. And so one of the I think, hallmarks of the story is we do have a few candidates here that could be old enough to be part of that original stocking, but the aging technique, it's going to be possible to say with true precision what exact year something was born at with this technique, and we really don't know all the information about when things were stocked and how many fish were stocked. And so we can't say that, but I'm sure some of the anglers would love to go out on a limb and do that. But essentially, this is a remarkable story that illustrates how much mystery there actually is in natural systems. And our ability to uncover some of that mystery, I think, is a really profound privilege. And to me, that is what drives me to do science. It's why I spent 5 years of my life doing it. And it's why I'm here. And so I find it to be a great privilege to be able to be a part of stuff like this. And I'm excited to do it here as well. And so with that, I can take questions about anything really. I think I've got plenty of time, so yeah.